The reason why we know so much about nucleic acid structure and function is because of the experiments by pioneering researchers. In this video, we're going to be talking specifically about experiments by Frederick Griffith and Avery, MacLeod, and McCarty in determining the transforming substance.
Until Frederick Griffith's 1928 study on pneumococcal bacteria, there's very little known about what was the heritable information of a cell. Griffith conducted his study on rats using S. pneumococcus, a bacteria that he had seen a lot of sailors killed by during his time in the Navy. When given a non-virulent, which we'll call NV, or a rough variant of this bacteria, the rats inoculated survived. However, when given the smooth variant of this bacteria, the virulent, which we'll call V, these rats actually died. So, after seeing this, Griffith decided that he would heat kill these bacteria and administer them into the rats. Griffith heat killed the smooth or previously virulent bacteria, and he administered them with a shot. When the rats were inoculated with this serum made from the heat-killed, previously virulent strain, it was found that they were actually not killed, and they survived. However, when heat-killed virulent bacteria were mixed with the non-virulent bacteria, and they were injected into the mice, it was actually found again that this resulted in death. Therefore, Griffith concluded there must be some form of transforming principle. This transforming principle, which we now know to be DNA, was the subject of extensive studies, which we'll discuss now. To establish the molecular identity of the transforming principle, MacLeod, Avery, and McCarty carried out an experiment using proteases, DNases, and RNases. What they essentially did was they took Griffith's experiment and they carried it out where the samples of the heat-killed virulent and the non-virulent strains were combined together and were placed with different enzymes that would digest certain molecules. At this point, they had the identity of the transforming principle narrowed down to either a protein, a DNA, or RNA. So they use DNase, RNase, and protease. Each column in this chart represents a different hypothesis about which of these molecules was the transforming principle. The first column we'll call D for DNA, the second R for RNA, and the third P for protein. What this experiment was looking at was
if an assay contains the enzyme which degrades whichever molecule the transforming substance is, then mice inoculated by this solution should survive.
They carried out each test where a minus sign means the absence and the plus sign means the presence of. So the first test they completed, there was DNase, there was RNase, and there was protease. And so this solution was mixed with the inoculation with the heat-killed virulent and the non-virulent strains, and they found that the mice survived. Again, they completed another one that contained DNase, but not RNase, and did contain protease. And again, they found that the mice survived. And finally, they removed DNase, and they just had RNase and protease. And when this solution was inoculated into the rat with the previous cocktail of bacteria, they found that the mice actually died. Therefore, they concluded DNA was a transforming principle.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.